Hi, everyone, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today we're talking to Heidi Meyer, who's assistant professor in the Psychological and Brain Sciences Department in the Center for Systems Neuroscience at Boston University. Heidi studies affective learning, behavior, and brain circuits underlying those things. I mean, affective with an A, not affective with an E. It's just my accent that makes them sound like the same word. And she studies how these behaviors and brain circuits change during adolescence. Research in her lab is currently focused on behavioral inhibition, and especially inhibition in fear and anxiety. I suppose that's what we'll talk about today. And I should say new lab, actually, because she's just setting up her own new lab at BU. Congratulations on that.
Thank you very much.
And thanks for joining us, Heidi.
Thank you for having me. I'm excited to be here.
So joining us today is Tony Burgos-Robles. Tony
Hello.
is our own expert on affective learning.
That's what they say, yeah.
And our all-purpose podcast friend, Matt Wanat.
Howdy.
And me, I'm your host, Charlie Wilson. So Heidi, it's an opinion shared by many that adolescents are emotionally volatile. And it is sometimes assumed that this must have a neurobiological basis, but it seems daunting to try to study it in ourselves. So what can we learn about this from studying experimental animals, and what do we already know about it from that study? Can we even decide what time in the life of an experimental animal is the analog of adolescence in humans?
Mm-hmm. Yeah, so I think we have been able to identify a period that we're deeming to be akin to adolescence in rodents. There's some speculation of whether, adolescence is a very social construct. There are certainly experiences that the human teenager has that a rodent is not likely to have. But there's a lot of benchmarks from the physical to, growth spurts, puberty, hormone changes that map on to a certain time frame in rodent adolescence that we also see in humans. And then there's also major differences in social and emotional behaviors. So there's this significant shift from a focus on the caregiver, the mother they've lived with for 21 days, 4 weeks in the case of a rodent, and then shifting to their conspecifics, so their peers, as we see in humans, this kind of focus once you enter middle school, high school, that gets you to focus more on your peers than your parents. Other things like exploring the environment more, maybe taking a few more risks. And all of these behaviors track. They're very similar species-specific versions of them, of course, in rodents, but really all kind of match up to the idea that adolescence is this time when you are working to sample the environment and preparing to go out on your own, preparing to be able to be interacting with new people, new environments, and kind of adapted to anything that you might encounter. And we see a lot of similarities in terms of rodents and humans in that regard. So in rodents, it's a much shorter period, which makes it easier to study than in humans. It's about 3 weeks. Postnatal day 30 to postnatal day 50 is the approximate range. So I think that's a huge benefit for rodent researchers, because we can get a lot of information about these rapidly changing brain circuits and these behaviors in a span of 3 weeks. We don't have to wait up to potentially close to a decade, as we would in humans, to see similar changes.
Does it map onto brain development? Like the human brain continues to grow through adolescence and then shrinks? Or does that happen in...
I think certainly there are markers that absolutely map on. So one of the probably biggest ones that many people have heard about is that the prefrontal cortex is continuing to develop throughout adolescence. It goes until the very end of adolescence, and sometimes even beyond. In humans, the prefrontal cortex, we know, continues to develop up until 25 years old. So that would be well after what we as society sort of deem to be the end of adolescence. And similarly in rodents, prefrontal is developing through late 50s. Late 50 days in some cases. And alongside that, there's also sooner development of other regions, so more emotional regions like the amygdala, like the nucleus accumbens. So that kind of leads to a lot of these canonical adolescent behaviors that we see, where you have this potentially increased emotional drive driven by the amygdala, driven by the nucleus accumbens, with slightly less input from the prefrontal cortex to tamp down those responses. So you get a lot of these emotionally charged behaviors. I do want to point out, though, that so adolescents, they kind of have a bad rap as being, risk-taking, kind of like this period where they're just hard to control, like what's going on there. But I think it's really important to say that the brain is actually hardwired for the experiences that they're having. They're taking more risks because they're exploring new environments. They have to learn new things in order to leave the nest in the case of a rodent, but meet peers, meet friends, learn things, get away from kind of the home environment, and prepare for independence. So a lot of the things that we deem to be risky or affectively charged are actually really just the adolescent doing exactly what they're supposed to do during that time. So I think that's my little soapbox that I have to stand on.
So a lot of what you were saying about the amygdala driving this and the cortex doing that, that's not based on human research. That's probably based mainly on animal research. So what's the nature of the evidence that you're
there's certainly both. There's parallels in functional neuroimaging. There's also, unfortunately, postmortem studies in humans that have allowed us to really kind of map the trajectory of brain region development. A lot of the more nuance of the structural connectivity and the functional connectivity, so how brain regions communicate with one another, how that changes across development is certainly easier to do in rodents because you can really get in there and interrogate circuits with techniques that allow you to actually record from very specific projections or specific cell types and increase their activity, decrease their activity in a very targeted way. So, but I would absolutely say there's a combination of both human and rodent and among other animals research that has led to what we know.
So what do we know?
Not nearly enough. I'm making a career out of studying this, so hopefully we don't know it all yet. So, if I can kind of jump into my own research.
Absolutely.
So, as I've sort of already alluded to, we have these regions that are potentially really primed to respond to emotionally significant stimuli. And this can be reward stimuli. This can be also fear stimuli. You mentioned anxiety disorders. Certainly in many cases, if your response to a fear stimulus goes into overdrive, you may experience an anxiety disorder. They're unfortunately very, very common, especially actually in adolescent populations. Peak, I think, diagnosis age is around 13 years old. So it's much younger than we would even want to think anxiety is becoming an issue. But my work actually kind of comes at that from a different perspective. So there is this notion that's founded by behavior that's also based on the brain, where adolescents are responding to fear stimuli very, very strongly. So they're exhibiting high fear responses, they're exhibiting anxiety. But my work comes in kind of from a different perspective of what about something adjacent to fear, or the antithesis of fear, something like safety. This can be emotionally charged. Safety can have rewarding properties. It certainly represents the absence of threat. So is that something that also could be emotionally charged in the same way? Might the adolescent brain actually be primed to respond to something that's explicitly safe in the environment in a way that the adult brain can certainly recognize and use safety, but there may actually be advantages to identifying and training adolescent mice and eventually teenagers to discriminate what around them is actually safe and use that to help regulate their emotions. So that's a lot of what my work is interested in, is recording from different brain regions under conditions of threat. So it's a stimulus. In our case, it's you play a tone for a mouse, you pair it with a mild foot shock, they become afraid of that tone, or you play a second tone that's never paired with a foot shock, so they learn that that second tone is safe. And we're able to then look at differential activity in brain regions to those two stimuli and kind of get at what is the seat of that form of safety in the brain.
Hey, if I can jump in, perhaps I can ask a silly question, but perhaps, for the folks out there that will see and listen to the podcast, what is the importance in the first place of having all these fear and anxiety signals, and on top of that, because they become problematic in a certain number of individuals in the population, then new functions of the brain need to be developed, like safety signals, in order to control those that are becoming problematic. Isn't better, or perhaps an error that happened in nature throughout evolutionary processes that spike these fear anxiety signals? I think so. Fear is actually very, very adaptive. There are tons of circumstances, even in your daily life, just kind of going out your front door, trying to walk across the street. You see a car coming at you too quickly. You sort of have that immediate fear response.
And that allows you to, engage a behavioral repertoire that allows you to stop at the crosswalk and not walk in front of the car. I think if you're out, I don't know, we're in Texas, so I don't know the examples like what you have on hikes, but let's say we're in the Northeast, we're out on a hike, you see something on the ground like kind of long and skinny, you're like, is that a snake? You want to be afraid for a moment because you want to run away if you need to.
Right.
So
this is the time of year
in Texas. But yeah, so you want to have that kind of immediate moment of just like being aware of your surroundings, having that like quick, like the fear system will provide this arousal that allows you to like act very, very quickly.
Right. Overall, like combining all these functions together, perhaps, like the overall umbrella function is behavioral flexibility so that you can adapt to any challenges in the environment in a manner that will keep you safe, will keep you, behaving just normally in the environment without even being panicking or so afraid because so many threats naturally occur in nature.
Absolutely, yeah. I think I would challenge, I guess, our viewers, our listeners, all of us to kind of, throughout the day, think of things that, like, for a moment, you have this moment of, like, oh no, like panic. Yeah. That's your fear response. That's a very adaptive fear response. But just then also notice how quickly it tamps down and you're able to just continue going about your business. So these are all very adaptive moments that allow you to go out into an unknown world safely. So you have all these like versatile experiences, but you're able to do them in a very safe manner. And that's the sort of baseline, like fear is adaptive, safety is adaptive. These circuits are absolutely in place. And then certainly I think the perspective that I come from is instances where maybe the fear is a little bit more than you would like it to be. And then in very extreme cases, anxiety disorders.
It's an intriguing thought, like an intriguing exercise that we all could do because we don't typically think about all the dangers that exist in our environments, and there's many if you really stop and think about all of them, right? Thing is that, like just reminds me of like the earlier work by, people like Joe Ledoux and other folks that determined or realized that all these threat-like fear signals even occur at unconscious levels, right? You don't really need to think about those in order to react appropriately to those dangers. And I'm wondering, like now that the field has been moving towards understanding other related functions like safety signals and how they interact with the threat signals, then I'm just asking, is actual consciousness required, like to come up with a response or giving an entire meaning of the entire situation with these more complex functions, or do we know anything yet about that?
So your question is
I guess like just because these are like the earlier times within this safety learning field, maybe not much has been done yet, especially like with humans. A human can tell you, I had to put a lot more conscious effort in order to come up with the best solution that will give me the most safety, but in animals it's harder to.
Well, in these experiments, you're not really asking your animal to overcome their fear, like to do something that they're afraid of in order to get a reward on the other side.
Right. The most beneficial thing. Yeah.
But instead, you give the animal like an easy choice, right?
Yeah. So in my task, it really is, and they get shocked either way, unfortunately. So it's a very controlled environment, and in that way, it doesn't recapitulate as much of the human complexity as additional experiments would, because there are very few instances in which sort of a fear stimulus, is forced upon you, something bad happens, and then another time a safety stimulus is forced upon you. In most cases, we do engage these cognitive processes. We do identify for ourselves, what do I know makes me feel safe? Potentially you carry something with you that makes you feel safe, or if you're in a new environment, or with a new group of people, you identify the person that makes you feel safe. You identify the part of the room that makes you feel safe, or stimuli that tells you, this situation is not threatening. But those are things that you are actually kind of accessing when you need them in the moment. You kind of identify them and then utilize them at that time. It's not like you have a fear response and then, a tone plays and you're like, oh, that car is not going to hit me because I hear this.
Instead, you see you're projecting the car's a trajectory, and at first it looks like it might be coming toward you, and then later you see that it's not.
Yeah.
And so that's pretty sophisticated calculation of the car's future position.
But even in these apparently simplistic models in animals, I think just the simplicity of the tasks kind of hide other processes that may be occurring, but we cannot actually quantify, or at least haven't come up with good solutions to quantify. Because, for example, like in similar tasks in which the animal is provided an option so that they can avoid or escape the danger, you would imagine you as an individual, as a human, to think about what if I do this, and make predictions of the near future, right? And that involves more cognitive engagement, and perhaps we don't.
But do rodents do that stuff?
What was that?
But rodents do that, don't they?
Yeah, that's what I would imagine. That a rodent would actually make those kind of predictions and integrate those in the calculation.
So in your experiments, you ask the rodent to do that.
But it's hard to quantify, and I guess like we as a field, perhaps should come up with some clever solutions to try to do a deeper understanding of all these processes, which may seem simplistic, but I believe from at least that they are more complex than what we.
I think that's also kind of like the benefit, the aim of a lot of the animal research is to break a very complicated behavioral repertoire or emotion down into its component parts. And so, I mean, I certainly study one aspect of safety. You study another aspect of safety. I think we've talked about how you're also studying safety, but potentially manifesting in a very different way. And so, like, what is safety? That is a very complex question.
So one of the things, I guess, if you could probably touch on for those who are not familiar with your work is, how you are sort of trying to use the rodent model and, potentially bridge that to sort of translationally relevant, issues. I mean, I think something we've sort of been scooting around is, fear can be adaptive and good, but it can also be problematic in the case of PTSD. And so how can we use, potentially safety signals to be able to reduce that, pretty much strong association of a cue that's no longer associated with a bad outcome. And I mean, some of your work really elegantly sort of gets at potential strategies that might be able to be used to weaken that association. I guess, could you sort of talk a little bit about
definitely. I think what the sort of immediate thing is that the form of safety that I'm studying, there is this moment in time where the safety signal is presented, the mouse is able to inhibit their fear kind of in that moment. And what's nice about that, whether it's an externally played tone, like in my experiments, or a human who has identified their own safety signal, it allows you to kind of have a moment of solace where you can reevaluate your surroundings. So when you see something that you do deem to be threatening, it actually, like, all of your attentional resources are employed to that thing. So it actually makes it more difficult to look around and say, what else in this environment might tell me that I'm safe? So if you have something where you can automatically say, like, let's bring down this physiological response, let's give us a moment to identify, is this a threat? How likely is it that this will actually cause something bad to happen? Let me look around, let me engage other cognitive processes. So I think that's one way where something like a safety signal can potentially have strong value, is just it gives you a moment to breathe and to think and to remind yourself that, like, yes, I am capable of coping with this situation. And another study that we have is to use, it's an extinction paradigm in rodents, but it has some similarities to exposure therapy in human clinical settings. So basically, extinction as a model is something that you have been afraid of in the past, or perhaps a tone in a mouse that's been paired with a foot shock is played repeatedly in the absence of a foot shock. So over time, the mouse will learn this tone no longer predicts a shock. I should not be afraid of this. This is a very effective means to reduce fear responding. There are some limitations over time. The fear response can return. So what we've been playing around with is how can you modify that training, this extinction training, potentially introducing something like a safety signal to help make that learning stronger and make it so it's a little bit more long-term. So one of our experiments actually compared, mice underwent regular extinction training, several fear cues, no shocks. And then another group where we took out half the fear cues and replaced them with safety cues. So they do have half as many fear exposures. They also now have these intermittent safety cues. And we did see that the memory later on, the reduction in fear actually was maintained for at least 2 weeks in the rodent life. So it's kind of this idea of, again, can you have these emotionally salient fear-producing events, but kind of couch them amid experiences that allow you to tamp down that physiological response, feel safe, experience your safety signal, and might that actually lead to stronger extinction learning or stronger fear regulation across multiple domains?
So to sort of follow up on that, and I mean, I'm talking out of my beep here, but so the safety signal that you're using is an intrinsically neutral stimulus. So it's just another tone that, really the animal is associated, I'm not going to have a bad thing happen now. And I guess has anywhere either in the preclinical or in the clinical research sort of thought, well, what if we give something good?
Yeah.
Would that potentially be something that could, hey, I'm hungry and get a food pellet? I know a lot of these studies aren't necessarily done under food deprivation, but is there potentially a way that you could have a rewarding stimulus? And so instead of it's just a safety cue, that's neutral, what if we had a positive thing that was embedded on top of this cue would that sort of serve to weaken, or facilitate extinction and weaken the initial sort of association between the cue and the aversive outcome.
Right.
And again, talking completely out of speculation here, but it's curious if we want to think about taking these preclinical models and translate it, bench to bedside, if you will.
Back in the day, counter conditioning, right?
Mm-hmm.
Kind of.
Yeah, I mean, that is a really good question. I think counter conditioning is a good example. So pairing something formerly that was paired with something aversive is now actually paired with something positive or rewarding. I'm not entirely sure about the extent to which you could just replace a safety signal with something rewarding.
But it's to embed something maybe on top of that. So It's now instead of just tone, but a tone that gives, hey, a little bit of juice, and something that would be intrinsically rewarding. Is that something that potentially could facilitate, I guess,
See, to me, Matt's experiment makes sense in terms of learning theory, like classical learning theory, whereas your result doesn't. I mean, here's just another thing that's happening during extinction. It's completely unrelated. That other tone has no meaning, really, at all. And yet, it facilitates extinction.
Right. So I will pause you there and say that I don't think at this point, in this point in our training, that we actually have a neutral stimulus anymore. Because the reason that I call it sort of a safety signal and not just in the technical jargon, it would be a CS minus, a conditioned stimulus that doesn't have value essentially, because we have trained it so much, and we really do overtrain these mice. They have a lot of experiences that contrast this safety cue with fear. So it's not only a neutral cue, it is actually something that's explicitly safe. And I think that's what's key.
So if you just pulled a new stimulus out of a hat, it wouldn't work, you think?
Potentially, but then I also showed a study today from Joey Dunsmore that shows that if you do have something novel in place of an aversive outcome, you actually do see similar benefits. So I think there's a lot.
It's intriguing to me, and I actually have a similar question, perhaps to grill you on this, why do you really think it's a safety signal, and perhaps not just, a meaningless, cue in this case? Because, to me, at least in the paradigms we use in my lab, animals are given the chance to experience something threatening, dangerous, and then they can do the behavioral correction because they already understand the meaning of the cue, right? And in that case, I would believe, you need to understand first that something is dangerous, but no longer is dangerous in order to give a safety meaning.
Yeah.
Is there any way you can disentangle that from your paradigm?
Yeah, I think the test that we do where we give the mice both options, they have a fear cue and a safety cue played at the same time, and we see a reduction in fear in that instance.
Right.
I think that's kind of that's our test for, it's a process called conditioned inhibition for any of the learning theory nerds in the audience. But that's really our critical test, because up until that point, it is just it's contrasted with fear, but we don't have any proof that it is anything more than just a neutral stimulus or a not a not fear.
Right.
So that's our test to really say that it has these properties that are capable of reducing fear, even when you have the fear cue present.
I like that explanation. It makes so much sense. But I'm wondering then why isn't the aversive meaning of the CS plus being transferred to the CS safety minus when you combine the two?
Well, so
Isn't
I think it does sometimes.
Yeah.
I mean, I know I don't want to misquote, so I'm not going to say a name, but there is someone who has done work, and I think it was specifically in females, where they actually showed higher freezing to a safety signal after it had been in compound because there was this transference of fear. So it kind of can go either way. So that, I mean, in a controlled laboratory setting, that is something that you're risking. You're starting from a neutral situation, and you have this, it has developed safety properties through training, but it can potentially just as easily lose those. So that's why if we're thinking of, therapeutic applications, you'd want something that's a little bit more robust, that you have more experiences with to really like amass that and secure that safe properties, and also potentially the transference of safety into the domain of being rewarding. And there is some evidence to show that many safety signals also do activate parts of the brain that respond to reward.
So I don't want to let you get away without talking about adolescence. So I was just thinking, the kinds of experiments you're talking about right now are, what's the difference between an adult and adolescence along all of those phenomena that you just described, the extinction and the...
Yes. So the biggest difference is probably in the ability of a safety signal to facilitate extinction. Up until that point, discriminating fear from safety is very, very similar between adolescents and adults. Even pairing them together for conditioned inhibition, that's also very similar between ages. But there's something about then pulling out that safety cue and applying it in new situations, like putting it in with extinction or trying to train that safety cue to be aversive. So if you pair a shock with that safety cue, both of those processes tend to be more robust in adolescence. The safety properties of the safety cue actually persist to a greater extent than they do in adults. So we'll see a stronger benefit of less fear recall later on. In an adolescent who's had a safety signal intermixed with extinction, we'll see slower fear learning about a safety signal paired with a shock in an adolescent relative to an adult. So we think there's something just inherent about the way that the safety signal itself is represented in the brain that may actually be a stronger neural signature than during adulthood, which kind of relates back to how I started into all this, which is the adolescent brain is hardwired to be responsive to emotional stimuli, so perhaps the safety signal falls into that category.
So that's a kind of developmental thing you might see in the brain. You can look for that, right? I mean, you have a handle on what kind of experiment you could be. I'm asking you, can you find a difference in some activity in some part of the brain or anything like that?
Absolutely. So some of the work that my lab is doing is looking at a part of the brain called the Ventral Hippocampus and its communication with the prefrontal cortex, and specifically the prelimbic region of the prefrontal cortex. And this circuit is structurally more connected during adolescence in mice. This has been shown, not yet shown in humans. And B, responds more. There's higher neural activity in this circuit during presentations of a safety cue during adolescence than during adulthood. So it kind of goes along with what I've been saying about how there may be this potential that the brain is actually showing a heightened response to this condition of safety during adolescence. And perhaps because that's just the way it's built. Basically, it's structurally wired to be more responsive to something like a safety signal.
So this is a naive question, but I mean, if, as sort of you've been framing it that, juveniles and adolescents need to sort of be able to sample a lot and they need to figure out what's good, what's bad. Do they exhibit sort of faster acquisition of, fear-related behaviors, or, I guess it's a difficult question to get at, but could smaller intensity foot shocks, can that give rise to sort of a higher level of freezing in adolescents relative to adults, knowing the fact that they're sort of, they might be feeling it more, less, fat and all that to sort of, numb the shock. But I guess, do we see faster learning in adolescence? In many cases about individual cues. So a tone that's paired with a foot shock or a tone that's paired with a food pellet leads
kind of one-to-one associations, either rewarding or aversive, you do actually see stronger, steeper learning curves. So they acquire, a freezing behavior faster. Fear conditioning is faster in adolescents than it is in adults. They will press a lever more or run to a food cup more to collect a food pellet when a tone plays than adults will. So they have very strong initial learning about these emotionally salient stimuli.
Another of your interests is the sex differences, right? And from the standpoint of on clinical perspective, females, the proportion of females suffer anxiety diseases are twice as many, right, as men. And I'm wondering, what kind of differences perhaps in this ventral hippocampal prelimbic circuit you may encounter or you're expecting to encounter that would explain this sex difference.
I don't have an answer for that right now. I think because we're only seeing I spoke earlier today about how the sex differences issue needs a lot more work done, because there's differences between rats and mice right now. There's kind of differences between how safety is trained and how fear is trained, and we're seeing sex differences in some places and not in others, and I'm seeing potentially sex differences in adulthood but not in adolescence. So there's so much more to unpack there before I could but you're absolutely right that anxiety disorders, psychiatric disease in many instances is different between the sexes, and so it's so important
we'll have you back. Let us know when you're ready to give us an answer to that one.
And I guess a related question also is that you briefly touched today was to induce these disease states by integrating prolonged stress paradigms. And my guess is that
we're not going to have time. Sorry.
Stay tuned in my lab.
Just to finish the thought, I guess, her research will be becoming more and more intriguing as, she keeps progressing in all these aspects.
So
thank you.
Your new lab has a lot to do.
Yes. Oh my gosh.
Thanks for joining us today, Heidi.
Thank you.
And Tony and Matt. And this has been Neuroscientists Talk Shop.